Hello. Can you hear me?
Yes, ma'am.
Yes, ma'am.
Okay. So, we are talking about coenzymes. So coenzymes, it is loosely bound with an enzyme molecule. So when the reaction with chemical gets completed, it will be released from that apoenzyme so that it can bind to another enzyme molecule. And we know it is said that one molecule of coenzyme is able to act upon a large number of substrate molecules with the help of the enzymes. And these coenzymes actually can be divided into two groups, and most of the coenzymes are derivatives of vitamin B complex substances. Okay, what are vitamin B complex? It's chemistry. You will all learn in your future classes. So just remember this. These coenzymes are derivatives of vitamin B complex substances. So it can be divided into two groups. So first group of coenzymes, it takes part in reaction catalyzed by oxidation and reduction, that is donating or accepting hydrogen atoms or electrons. So these coenzymes take part in oxidoreductases, oxidation reduction coupling reactions. And the second group of coenzymes take part in reactions transferring groups other than hydrogen. So the first group takes place in reaction transferring or accepting hydrogen atoms or electrons, and the second group of coenzymes than hydrogen. So these are the two group of coenzymes, and most of the coenzymes belonging to these two groups come under vitamin B complex substances. So first group of substances, as I said, it will transfer or accept hydrogen atoms or electrons. So for examples, the first group of coenzymes are NAD, NADH. NAD is nicotine adenine dinucleotide. Nicotinamide adenine dinucleotide. Okay, these are derivatives of vitamin B complex. NAD, NADH, NADP, NADPH, FAD, FADH2, FMN, FMH2. These are the examples of first group of coenzymes, and these are all vitamin B complex. You will all learn what are these in your vitamin classes. Okay. So this is otherwise known as this first group of coenzymes, this NAD, NADP, FAD, FMN is otherwise known as co-substrates or secondary substrates. So if they ask you to write a short note about co-substrates or secondary substrates, they are intended to write what are coenzymes and what are the first group of coenzymes. Okay. Now, see, are you familiar with this reaction?
Yes, ma'am.
Yes, this is actually where is this happening? Where is this reaction happening?
Glycolysis.
Breakdown of glucose, glycolysis. What? Like, so glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate by the action of the enzyme glyceraldehyde 3-phosphate dehydrogenase enzyme. So here the co-substrate is NAD. So NAD is actually the hydrogen atom liberated from the substrate is accepted by the NAD, converting into NADH. Now, can you remember this part? What happens when in anaerobic respiration? Yes, anaerobic respiration, pyruvate is converted into lactate. So the NADH that is formed in this reaction, it is used up during anaerobic respiration. And this hydrogen atom or electrons is given up during those reaction to convert the molecule of pyruvate into lactate by the action of the enzyme lactate dehydrogenase. Okay, so this is an example of first group of coenzyme. Now, what is second group of coenzyme? As I said, it helps in reactions transferring groups other than hydrogen. So these all belong to, these are all actually thymine pyrophosphate as vitamin B1. So it actually helps in the transfer of group hydroxyl ethyl. Okay, you have to just remember only those names. We don't need any structures here, right? So which group is transferred by thymine pyrophosphate in an enzymatic reaction? It is hydroxyl ethyl. That only you have to remember, right? We don't need any structures here. We only need structures in molecular biology classes and just carbohydrate, lipid, and proteins basic chemistry classes. Okay, in enzymes we don't need much of the structures. So just remember those names. In which group is transferred in thymine pyrophosphate is hydroxyl ethyl. PLP, pyridoxal phosphate. The group transferred here is amino group. Then biotin, it is also a vitamin B complex, right? The group transferred is carbon dioxide. Then coenzyme A, it transfers acyl group. Tetrahydrofolate or folic acid, it transfers the one carbon group. Just mention all this. You will get an idea once the class progresses, right? When you complete the protein and the lipid metabolism and all, you will get a good picture about this. Just now listen what I'm talking. Okay. Now, adenosine triphosphate, you know that ATP, it's a universal currency of energy. It transfers the phosphate group. Can you give me an example where you have studied an enzymatic reaction where you studied yesterday where ATP is used as a coenzyme?
Glucose is converted to glucose 6
reaction of
phosphate.
glycolysis.
First step.
Okay, very good, very good. But you all know it, right? How the ATP is donating the phosphate molecule, converting the substrates to another product. So these are the second group of coenzymes. So coenzymes can be classified into two groups. First group of coenzymes which donate or accept hydrogen atoms or electrons. Second group of coenzymes transfer groups other than hydrogen. For example, these are the examples, right? Now, we are talking about the compound enzyme. A compound enzyme consists of a protein part and a non-protein part. So the non-protein, we are talking about more about the non-protein part. So the non-protein part can be a prosthetic group, and it can be a coenzyme, or it can be metal ions. If the metal ions is present as the is combined with the apoprotein part to form the holoenzyme, that enzymes are known as metalloenzymes. Okay, so these enzymes actually require metal ions for their activity. For example, tyrosinase. This enzyme actually require copper as a metal for its activity, and it is some enzymes, some metals will be tightly bound and some it will be loosely bound, but when the enzyme gets active, the metal will be that is when will the enzyme be active? When the metal ion is added, so the activity of that enzyme will be enhanced, and that group of enzymes is known as ion activated enzymes. For example, it's a pancreatic lipase enzyme, okay, which break down the lipid. So it requires calcium ions. When calcium ions come and bind to pancreatic lipase, its activity gets enhanced. So that group of enzymes is known as ion activated enzymes. So metalloenzymes means it requires metal ions for their activity. Examples are tyrosinase enzyme. You will learn tyrosinase enzyme in protein metabolism, right? And another enzyme is pancreatic lipase. Tyrosinase requires copper and pancreatic lipase requires calcium. And ഇത് തന്നെ എഴുതണമെന്ന് ഒരു നിർബന്ധമില്ല. നിങ്ങൾ ബയോകെമിസ്ട്രി ക്ലാസ്സസ് നിങ്ങൾ മുന്നോട്ട് പോകുന്ന അനുസരിച്ച് ഒരുപാട് എൻസൈംസിന്റെ പേരുകൾ പഠിക്കും. ആ എൻസൈംസിൽ മെറ്റൽ അയൻസ് റിക്വയർ ചെയ്യുന്ന ഒരുപാട് എൻസൈംസിന്റെ പേരുണ്ട്. ഏത് വേണമെങ്കിലും നിങ്ങൾക്ക് എഴുതാം. ജസ്റ്റ് ഫോർ എ ഇൻട്രൊഡക്ഷൻ സേക്ക് ഞാൻ ഈ എൻസൈംസ് പറഞ്ഞതേ ഉള്ളൂ. ഓക്കെ, so these are the enzymes and the metal required for those enzyme activity. Okay, now what is cofactors? Actually, cofactor means it is actually in your textbook it is given as we can, what do you call it? It's a collective term to include both the coenzymes and metal ions. It is known as cofactors. Coenzymes can be otherwise termed as cofactors, or metal ions can be otherwise termed as cofactors, and sometimes it is collectively called as cofactors. Okay, So certain enzymes require certain cofactors. So it may include a coenzyme and a metal ion. So that type of that is known as cofactors. Now, yes.
Ma'am, will the prosthetic group be included in the COFACT?
Actually, in textbook, prosthetic group is not mentioned. Actually, it is a bit more complicated when you refer to highest textbooks. So you don't need to know that much actually. So I have mentioned only that it's that much needed. And cofactors is actually prosthetic group can also be included. All together, we can term it as a cofactor. Actually, it is not very much clarified in your Vasudevan Sri Kumari Madam textbooks, but you guys actually don't need that much of elaboration in this. Just study the basics only. Okay. Now we'll come to enzyme nomenclature. So, see, a lot of enzymes were discovered during the and they were given a trivial name, that is a practical name, which will be very suitable for everyday use. But Enzyme Commission, an organization was found out, and it was known as International Union of Biochemistry and Molecular Biology, IUBME, and they provided a name, and that name is known as a systematic name that describes the name, and that name of a particular enzyme describes the nature of the reaction catalyzed by that enzyme. And this commission has given an enzyme a unique numeric code. So IUBME classification of enzymes is important for you from exam point of view. It is last year university question was it was the university question. So this part is actually important. So they have given a numeric designation for each enzyme, and it consists of 4 numbers. And before the numbers, it is prefixed by the letters EC. EC means enzyme commission. And actually, enzyme commission, the 4 numbers are there. The first number denotes a class, and all enzymes are assigned to one of the 6 classes. There are total 6 classes present, and it is characterized by the type of reaction they catalyze. We will see. And the first number denotes the class to which the enzyme belongs. The next number, the second number, denotes a subclass, and the third number denotes a sub-subclass. And the last number, or the fourth number, is a specific serial number that is within a sub-subclass. Okay. Now, so coming to class 1. Class 1 is oxidoreductases. So this group of enzymes, as the name itself, you can write it. It will catalyze oxidation of one substrate with simultaneous reduction of another substrate. This can be generally represented as by this equation, that is like H2 plus B gives A plus B H2. You can write the reaction that has been shown earlier, right? In the first group of coenzyme, glyceraldehyde 3-phosphate converting into 1,3-bisphosphoglycerate. That can be also be written as an example for oxidoreductases. Now, here I've given another example. Alcohol. Alcohol is acted upon by the enzyme alcohol dehydrogenase to convert it into a lesser toxic molecule aldehyde. And here the hydrogen atoms are accepted by the coenzyme NAD, nicotinamide adenine dinucleotide, converting into NADH. And the IUBME has given this name alcohol dehydrogenase as alcohol NAD oxidoreductase. This is alcohol dehydrogenase is a trivial name. And the code number for this is EC1111. That is, it's a first enzyme. It belongs to class 1 oxidoreductase. It belongs to the subclass 1, subclass 1, and it is the first enzyme in that subclass. Okay. This oxidoreductase is also not only it oxidizes a hydrogen atom, it also oxidizes substrates by adding oxygen. Like oxidases, oxygenases, and dehydrogenases all belong to this class, oxidoreductases. So this is class 1 oxidoreductases. Now, class 2. It is very simple actually. You can write the definition from the name itself. Just remember the order of the names. That is important. You have to write these classes in the order and write the correct name. Transferases. Transferases means what? Transfer, right? It will transfer groups other than hydrogen, right? So you can here write an example. See, glucose combines with glucose will be converted into glucose 6-phosphate by the action of the enzyme exokinase. It requires one molecule of ATP, and the phosphorylation of glucose is where is the phosphate coming from?
ATP.
Yeah, ATP is donating that phosphate, right? And phosphorylation of glucose molecule occurs and converting into glucose 6-phosphate. So one molecule of phosphate is low, so it converts into ATP will be converted into ADP. And again dinucleotide phosphate. Okay, the enzyme is hexokinase. So transferases, it transfers group other than hydrogen. Okay, oxidoreductases, you know oxidoreductases now. Now class 2 is transferases. You can write any example, or like any example means this is not the only enzyme where transferases is taking place. You can write any other enzyme, so but it should be under the correct class. Now, class 3 is hydrolases. Hydrolases, we know that it hydrolase. Hydro means water. So by adding a water molecule, the substrate will be cleaved or it will break the bond. So it can hydrolyze ester bond, either peptide bond or glycosidic bonds. So a water molecule will be added. The example given is acetylcholine is converted into choline plus acetate. That is breaking down of acetylcholine into choline and acetate by the action of the enzyme acetylcholine esterase or acetylcholine hydrolase. It is the systematic name. So this belongs to the class hydrolases, and we have to know that all digestive enzymes inside our body belong to hydrolases group. Okay. Now the class 4 lyases. So lyase means what? It's also breaking the bond. So lyase, it means cut. So it will remove the groups or atoms from substrates so that it can break the bond, but it is not by the mechanism of hydrolysis. That is not by adding water. An example given is, you will be familiar with this reaction, right? Fructose 1,6-bisphosphate is converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the action of the enzyme aldolase. So aldolase is actually enzyme is what is doing it as. It belongs to the class lyase. So it is breaking down the fructose 1,6-bisphosphate molecule into one molecule of glyceraldehyde 3-phosphate and one molecule of dihydroxyacetone phosphate. Now class 5, it is isomerases. Isomerases, as the name itself, it produces optical or geometrical or positional isomers of substrates. An example here is, it's all examples given from glycolysis. Okay, glyceraldehyde 3-phosphate converting into dihydroxy. These are isomerase by the action of the enzyme triose phosphate isomerase. Now the last class is ligase. Ligase means to link, to bind. Latin word ligare, to bind. So it will link two substrates together, but by simultaneous hydrolysis of ATP. It requires a molecule of ATP to link two substrates. This example is actually the first step in lipid synthesis. Okay, how fats are synthesized inside the body. So this is the first step inside in that lipid synthesis. You will learn in your future classes. One molecule of acetyl-CoA combines with one molecule of carbon dioxide to form malonyl-CoA. This is the first step in lipid synthesis. An ATP molecule is required, and it is donating a phosphate molecule, and it's converting into ADP, and that phosphate will be released. The enzyme here is acetyl-CoA carboxylase. Ligases require ATP. Okay. Now there is actually code for you to remember these 6 classes. Anybody know that? It's actually sorry. Okay, is this? Okay, that's why we used to remember like this OTHIL, like this is OTHLYIL. See, you have to write the classes in their order itself. Then only you will be given full marks. So IUPME classification of enzyme is class 1 is oxidoreductases, class 2 is transferases, class 3 is hydrolases, class 4 is lyases, class 5 is isomerases, and class 6 is ligases. Okay, you have to write in this order itself, then only you will get good marks, means full mark, and you have to write an example. Okay. So actually I said ligases. Ligases require ATP. So you will be hearing a lot of name of enzymes. So some enzymes will be synthetases, like ending as synthetases, and some enzymes will be ending as synthases. So what is the difference between synthetase and synthase? So synthetases require ATP, and they belong to this class ligases. But synthases also it also catalyze biosynthetic reactions, but they do not require ATP directly. They belong to other class that is other than ligases, other than the classes. Examples are also given here. You will learn in your future classes the difference between synthases and synthetases. Okay, just I mentioned it. What is the difference between synthetases and synthases? Synthetases requires ATP, and synthases don't require ATP. Okay. Now, we have completed the classification of enzymes. So we are coming to some details about an enzymes. So what do you mean by an active center of an enzyme? So we will be hearing this active center in later of these classes. So first we have to learn about what do you mean by active center of an enzyme. So an enzyme consists of a particular region where the substrate go and bind, or the actual reaction taking place. And that part of the enzyme is actually known as active site or the active center of that enzyme. Almost all parts are required for maintaining the three-dimensional structure of the enzyme, but the reaction will be taking place only at that active site, and it will occupy as only a small portion of the whole enzyme, and it will be like hidden. It will be situated in a crevice or a cleft of the whole enzyme molecule. And what are the properties of this active site? This active site is specific substrate bound. That is, active site, the specific substrate only can come and bind to this active site. No other molecule can come and bind to this. And it depends upon the alignment of atoms or the amino acids that is present at the active site. You will get more clarifications later in these classes. Okay, so and what happens to this? Sometimes one substrate come and binds to the active site, it induces a conformational change to the active site. That is, it will produce a change at the active site so that the substrate can actually bind to it, bind correctly to the active site. Okay, and it binds to this active site by non-covalent bonds. Are you able to hear me? Hello?
Yes,
Yes,
ma'am.
Yes, ma'am.
Yes, ma'am.
Are you able to see my slides?
Yes,
Yes, ma'am.
Is there any disturbance?
No, ma'am.
Ma'am.
Okay, fine, thank you. Now, and these substrate will bind to the active site of the enzyme by non-covalent bonds, and these forces are hydrophobic. You know, of course, what's the meaning. And now, there are, as I said, at the active site, there are specific amino acids or groups will be there, and that will help in the substrate to bind to that site. And these specific amino acids or the groups that are seen at that active site of an enzyme are known as the catalytic residues or the catalytic groups where the catalysis is occurring. Okay, now the active site actually contains two separate sites, a substrate binding site and a catalytic site. That is, once a substrate comes and binds to the substrate binding site, then only the catalytic site gets activated and the catalysis occurs. Sometimes it will be same. So for some enzymes, and for some enzymes, there will be two separate sites will be present. Okay. Actually, I can just hold on. I might actually finish that.
सुन लिया? सुन नहीं रहे? पढ़ो।
Yes, are you able to hear me?
Yes, ma'am.
Just 1 second. There's actually some problem here. Yes. So the active site means it is the region of the enzyme where the substrate binding and catalysis occurs. Sometimes there will be two separate sites for that. Sometimes it will be the same. So now coming to how these enzymes work, or what is the mechanism of action of these enzymes. And actually this has been explained by a lot of theories. So the first and the foremost important theory that is needed for you actually is this first one, lowering of activation energy. That is, enzymes work or act by lowering the activation energy. So these are the other theories explaining how the enzymes work. Actually, these are all not needed. I'm just mentioning the names only. Acid-base catalysis, substrate chain, covalent catalysis, entropy effect, product substrate orientation theory. These are all actually other mechanism or other theories explaining how enzymes are acting in our body. The important theory that you have to mention is this: how the enzymes work by lowering of activation energy. And this has been explained using three theories, that is Michaelis-Menten theory, Fischer-Template theory, and Koshland-induced Fed theory. So this is important for you, right? So how the enzymes work, right? By lowering their activation energy. So we're going to learn that. So we know that what like see what will happen if we want to burn something, right? We want to make a fire, or we want to keep a flame, and we will get some wood and we will try to burn it initially. Will it burn all on a sudden?
No, ma'am. You need the initiation.
We need the ignition temperature.
Yes, you're right. Absolutely right. Like it needs some time, right, to reach that to completely burn that wood, right? Then only the proper fire will come out. So for what will we do as a to make it simple? We'll add some like some kerosene or paper or something like that. We'll put it so that it will do that work very fast, right? So similarly, enzymes also will make the reaction faster. That is, see, this is a substrate, and this is the time taken for that reaction to take place without the action of an enzyme. So it requires a lot of time, right? So what will happen if we add an enzyme to the for the substrate to be changed into the product? It will require only a shorter time, and it requires a less energy. So activation energy means it is the energy that is required to convert all the molecules of the reacting substance from the ground state to the transition state. That is, enzymes will actually, what will happen? It will lowers the energy of activation. It will lowers the energy of activation. So this is how most of the enzymes work, and this has been explained using 3 theories. So the first theory is Michaelis-Menten theory. Michaelis-Menten, what did they say? Michaelis and Menten put forward the theory enzyme substrate complex theory. What will happen? See, So here is our substrate. So when an enzyme is added to a substrate, the enzyme has got a specific site, or the substrate binding site, and to that substrate binding site of the enzyme, it binds and forms the enzyme substrate complex. Once the enzyme substrate complex is formed, the active center of the enzyme is activated and the catalysis occurs. That is, the enzyme acts on the substrate and it is converted into the product. The substrate will be converted into the product, but it is not released from the enzyme. It forms an enzyme product complex. And finally, once it is completely formed, or once the reaction is completely over, the products will be released from the enzyme. Okay, so this is the theory that has been put forward by Michaelis and Menten in the year 1913. And this theory is known as enzyme substrate complex theory. So what is the basis of this is that we have learned just now. It will lower, the enzymes will lower the activation energy required, or it will reduce the energy required to convert the substrate to the product. Okay, this is the first theory explaining how the mechanisms work. A mechanism of enzyme. Now, next theory is Fisher template theory. So Fisher is another scientist. He proposed another theory like similar to a lock and key model. So here the substrate will fit to an enzyme. Like see, the enzyme and substrate will be complementary to each other. Only a particular substrate will come and bind to a particular enzyme. Not every substrate will come and bind. And the active site of the enzyme, at the active site, the substrate binding site will be actually complementary to the substrate. It's similar to a lock and key. So that the lock can be opened only by its own key. Likewise. So actually this theory was not very well accepted because it proposed a rigid structure for the enzyme, but actually enzymes are not rigid. They are highly flexible. And this theory does not explain the flexibility shown by the enzyme. So it was not accepted by the, not accepted. Okay. So actually Fisher template theory states that the 3-dimensional structure of the active site and that substrate binding site or the active site of the enzyme will be complementary to the substrate which comes and bind to them. Okay. Now the third theory is Koshland's induced fit theory. Koshland explained very well about the 3-dimensional structure of the enzyme. That is, the substrate will come and bind to the enzyme. Once the substrate comes and binds to the enzyme, it causes a conformational change at the active site of the enzyme, so that the substrate can actually very well bind to that specific part of the enzyme and the catalysis can occur. So that is the conformational change that is happening to the active site of the enzyme very well explains the 3-dimensional structure of the enzyme. So this theory proposed by Koshland was very well accepted. That is, what did he propose? He proposed that the substrate will induce a conformational change in the enzyme so that the substrate can very well fit into the substrate binding site, and the catalytic site is also getting activated so that the enzyme catalysis will take place. And this theory very well explains an important concept of enzyme inhibition, that is the allosteric inhibition, which we will be dealing in the next class. So this is how the enzymes work, or the mechanism of action of enzymes by lowering the activation energy is explained, very well explained by these three theories, that is the Michaelis-Menten theory, the Fischer's template theory, and the Koshland's induced fit theory. This part is actually very important from exam point of view, because as I said earlier, last year, your previous batch university exam question, essay question was IUBME classification of enzymes and describe the factors affecting enzyme action and mechanism of action of enzymes. So it's actually very important. So this explains how the enzymes work. The theories that explaining how the enzymes work by lowering the activation energy. For Michaelis-Menten's theory, put forward the enzyme substrate complex theory, then Fischer template theory, and the Koshland's induced fit theory. So I think I will stop here today. I want your feedback actually. How was this? Did you understood anything?
Yes, ma'am.
Yes, ma'am.
Okay.
Ma'am, many things we
It's a
have
bit
actually learned fast in
Speed
11th.
കൂടുതലാണോ ക്ലാസ്സിലെ സ്പീഡ് കൂടുതലാണോ
സൂപ്പർ ഫാസ്റ്റ്
Just a little bit, ma'am.
നല്ല ഫാസ്റ്റ് ആണ് മാം.
സ്പീഡ് കുറയ്ക്കണോ വളരെ കൂടുതലാണോ നോട്ട് ഏബിൾ ടു ഫോളോ
And we are
Just
able to
make it
follow
like
them, but we are not
correct.
able to write all things what you tell.
ഇനിയിപ്പോ അതങ്ങ് ശീലമായിക്കോളും വിഷമിക്കണ്ട
Same, ma'am.
അല്ല ശീലമാണ് നമ്മള് അത് ഇനിയിപ്പോ അങ്ങോട്ട് അങ്ങനെയൊക്കെ ആയിരിക്കും. ഫസ്റ്റ് ഇയറേ ഇപ്പൊ ഇങ്ങനെ ഒരു ക്ലാസ് കിട്ടുന്ന നിങ്ങൾ വളരെയധികം നല്ല രീതിയിലുള്ള ഡോക്ടേഴ്സ് ആയി പുറത്തു വരാൻ വേണ്ടിയിട്ടായിരിക്കും ഒരു പക്ഷെ. ഇനി ഫൈനൽ ഇയർ, സെക്കൻഡ് ഇയറും തേർഡ് ഇയറും ഫൈനൽ ഇയർ എന്നൊക്കെ പറയുന്നത് ഭയങ്കര സ്പീഡായിരിക്കും ക്ലാസ്സിനൊക്കെ. അപ്പം ഇത്രയും സ്പീഡിൽ ആരും പഠിപ്പിച്ചിട്ടില്ലടോ ഇതുവരെ അനാറ്റമി, ഫിസിയോളജി, വേറെ
പറഞ്ഞിട്ടുണ്ട്.
ഓക്കെ, ഐ ട്രൈ ടു റെഡ്യൂസ് മൈ സ്പീഡ്, ബട്ട് എനിക്കത് നടക്കുമെന്ന് എനിക്ക് യാതൊരു ഉറപ്പുമില്ല. ഞാൻ എന്തായാലും ഞാൻ ശ്രമിക്കാം. ഓക്കെ, നിങ്ങൾ പറഞ്ഞ സ്ഥിതിക്ക്. ക്ലാസ് അല്ലാതെ പൊതുവെ എങ്ങനെ ഉണ്ടായിരുന്നു? ലൈക് കുറച്ചെങ്കിലും ഫോളോ ചെയ്യാൻ പറ്റിയോ?
മനസ്സിലായി.
നല്ല ക്ലാസ് ആയിരുന്നു മാം.
എന്തെങ്കിലും സംശയം ഉണ്ടെങ്കിൽ ജസ്റ്റ് ഫീൽ ഫ്രീ ടു കോൺടാക്ട് മീ. ഒന്നെങ്കിൽ വിളിക്കാം, അല്ലെങ്കിൽ മെസ്സേജ് അയക്കാം, എന്ത് വേണേലും ചെയ്യാം. ഓക്കെ, എന്ത് സംശയം ഉണ്ടെങ്കിലും ചോദിക്കാം. I think this is enough. Actually, I think we'll see on next Friday.
Thank you.
Okay.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Thank you.